Hello everyone. Today we are going to present on our research project, which is effects of microgravity on human-induced pluripotent stem cells cardiomyocytes, and we decided to check the effects of microgravity on human cardiac and muscle activity. So, effects of microgravity. The effects of microgravity can be used to explain the improvement on the biological effects of radiation under the weightlessness condition. Microgravity is the most important influence on the human organism in space, and the long-term exposure of spaceflight undergoes dramatic changes on human body and results in severe osteoporosis and muscle atrophy, as well as neurological disorders. Microgravity induces many changes in specialized cells and stem cells, and it also helps in understanding and suppressing tumor metastasis or tissue engineering, where it induces the scaffold-free formation of three-dimensional multicellular spheroids. So we also gathered this pictorial representation from Canadian Space Agency, which shows like how does space affect the human body, and as you can see, it affects whole human body starting from brain, then heart and blood vessels, muscles and nervous system, bones, and it also damages the cataracts in the eye and damages the DNA, which increases the risk of cancer. Pluripotent stem cells. Human pluripotent stem cells, including embryonic stem cells and induced pluripotent stem cells, have the capability to self-renew indefinitely and to differentiate into derivatives of three germ layers that are ectoderm, mesoderm, and endoderm. Induced pluripotent stem cells are an exciting tool that offer the potential to advance the current state of the art, as they are derived from somatic cells, enabling the development of personalized medical strategies and patient-specific disease models. Induced pluripotent stem cells can be differentiated into cardiac myocytes, can form patches resembling native cardiac tissue, and can engraft to the damaged heart. And the first induced pluripotent stem cell were generated by reprogramming skin cells in the year 2006. Some of the studies found that induced pluripotent stem cells, cardiomyocytes, can couple efficiently to damaged heart and restore the cardiac contractility. Almost all found that induced pluripotent stem cell cardiomyocyte transplantation is arrhythmogenic, which is it causes beating irregularities in heart, thus hampering the use of induced pluripotent stem cell cardiomyocytes for cardiac regeneration. About our dataset, this is the GEU session number. Title: Effects of Spaceflight on Human-Induced Pluripotent Stem Cell Derived Cardiomyocyte Structure and Function. About the study authors did. So, exposure to microgravity was given to the stem cells, and they mostly studied on the uh, calcium handling of human-induced pluripotent stem cell cardiomyocyte and mitochondrial metabolism. Authors decided to perform the same study as previously done on mouse or rat. They generally focused on studying the calcium handling effect and the heart contractility effect. After the research, they concluded that they didn't observe a great difference neither in calcium handling or capacity nor in the heart contractility. So they suggested that microgravity affects mainly the mitochondrial metabolism, and this study can be used for further research. So this is the pictorial representation of GEU dataset page and sample characteristics. So the whole treatment and study was done for 4.5 weeks under the microgravity exposure, and after the exposure, the cells were kept for 1 week of rest. So the whole study was done on the basis of 5.5 weeks in total. So we had total number of 18 samples, 6 each in flight condition, post flight condition, and ground control. So we also created two different groups based on our samples, that is in flight versus ground and post flight versus ground. As the data on GEO was normalized, we collected the raw file from SRA, and then accordingly pipeline was run. So we first ran the RNA-seq pipeline, followed by quantile normalization, PCA, DESeq2, and enrichment analysis. So about the quantile normalization. Quantile normalization is a technique of mapping original values to the corresponding quantile of a standard distribution, and it gives the values to the data where null values are given after RNA-seq analysis, and we also did the PCA under this pipeline. About the DESeq2 analysis, we have taken the parameters for DESeq2 are like for log FC threshold -1 and P-adjusted value threshold is 0.05, as well as for GACA analysis parameters, P-value threshold was 0.1. And we also did the enrichment analysis and GACA analysis in this pipeline. Now I would like to ask Shubham to continue from here.
Thank you, Ojasvi. As we can see from the image that after the DESeq2 analysis, we have found the KEGG enriched pathways and GeoGACA enriched pathways, which were performed after the enrichment analysis along with the GACA analysis. These are the heat map results of ground versus flight normalized data and ground versus post-flight normalized data. From this plot, the regulation of genes can clearly be observed for each sample, that is ground and flight, in both ground versus flight and ground versus post-flight conditions. PCA, it is an unsupervised machine learning algorithm used for dimension reducibility and information compression. In our result, we can see that the sample-to-sample variation is dominating the PCA. Sample 2 and sample 5 in both flight and post-flight condition are clustering together irrespective of ground, flight and post-flight. The segregation of data is more pronounced in ground versus flight and as compared to ground versus post-flight condition. As it is clearly visible from the bar plot that the total enriched pathways for ground versus flight and ground versus post-flight for both GACA and KEGG, a great variation is observed. In ground versus post-flight condition, the enrichment of pathway was approximately double as compared to ground versus flight condition. Hence, there were a lot of differentially expressed genes and their statistical filtering was not giving the proper result. The data obtained from these pathways was statistically relevant but biologically irrelevant. Therefore, manual identification was made to filter the pathway for a further analysis. Because our project revolves around the effect of microgravity stress on cardiac and muscle cell, we have confined ourselves in filtering the pathway manually related only to cardiac muscle cells and stem cells. Similarly, a great variation was observed in the cardiac related pathways and in the muscle related pathways for both GACA and KEGG enriched pathways. After filtering out the pathways, we have sorted on the basis of the set size, E-score, p-value and p-adjusted values. From this, we have analyzed that first of all, authors focused on a very few pathways related to the study, that is calcium activity on heart contractility. They didn't focus on the other pathways which are important to the heart or cardiac related. They also didn't consider overall effect of microgravity, and also the stress was not observed. From our analysis, we have found two unique pathways. First is pathway related to pluripotency of stem cells in flight condition, and second is muscle related pathway in post-flight conditions.
So as Shubham just told that we have found the two unique pathways. So out of those two, one is signaling pathway regulating pluripotency of stem cells. So this pathway was found in the flight condition and it was suppressed in the post-flight condition. So what are signaling pathways regulating pluripotency of stem cells? The pluripotency of stem cells is regulated and maintained by transcriptional networks that include a series of pluripotent transcription factors. These pluripotent genes stimulate or inhibit the expression of downstream genes, including the occurrence of some signaling pathways and regulating the pluripotency of stem cells. The function of signaling pathways regulating pluripotency of stem cells retain the potential of self-renewal and multidirectional differentiation in the action of signaling pathways regulating the pluripotency of stem cells. So, having a better understanding of the mechanism of these pathways could help scientists study gene function and disease pathogenesis, and promote the development of regenerative medicine. So as you can see from the DESeq2 result, the highly activated diseases from this pathway are embryonal carcinoma and stomach carcinoma. Apart from that, we got 25 genes activated in this pathway, out of which the top 5 genes are HOXB1, FGFR4, HAN1, HESX1, FGFR3, followed by NUR and others.
Now the another unique pathway which was related to muscle was the vascular smooth muscle contraction pathway. It was observed in the post-flight condition due to the stress which was observed in the microgravity condition. This pathway is responsible for hypotension, hypertensive disease, and we can see from the DESeq2 result. And out of the 25 genes which were present in this pathway, highly expressed genes are ADCY6, ADCY5, and generally ADCY is adenylate cyclase 654, and other pathways and other genes are highly also responsible. This is the whole overall pictorial representation of the pathway of vascular smooth muscle contraction pathway. From this we can see the genes responsible and their functioning in the human body. This is the calcium dependent regulation of the vascular smooth muscle contraction, which was not observed by the authors. Generally, vascular smooth muscle contraction pathway is slow, and whereas cardiac muscle undergoes a fast and a relative short cycle contraction. Both the cardiomyocytes and the vascular contraction are dependent on the hydrolysis of ATP. Function of this pathway is to redistribute blood where oxygen consumption is temporarily increased. Excessive vasocontraction causes hypertension, and excessive vasodilation leads to the hypotension. In the conclusion, we want to say that the pathways enriched are statistically relevant but not biologically relevant. The cardiac pathways are not significantly changed in terms of flight and post-flight condition. Due to stress, change in muscle contraction was observed, and after 1 week of rest, cells didn't get recovered. They require more resting period. In ground versus flight and ground versus post-flight, enriched pathways were getting doubled in both KEGG and GAS analysis. We have also explained the two unique pathways which were observed in our research and was not observed by the authors. These were the unique findings from our research, which was totally outlooked by the authors. In the last, we want to say that our research work can be used for further study of effect of microgravity stress on a broad perspective for cardiac and muscle cell, and for study of diseases related to stress observed. Thank you.
Thank you.